So we have now seen that how during the process of digestion in the intestine, the different components of the different lipids like of phospholipids, of triglycerides and of cholesterol, how they are divided into their constituent components. So as a result, we have now these constituent components. What are those constituent components? They are free fatty acids, this is free cholesterol, and 2 MAG, that's a 2 monoacyl glycerol, which is in the jejunum, a part of the intestine. Now a very important thing happen here, that because lipids are mainly the hydrophobic in nature, major portion of all the lipids is hydrophobic, so it's very difficult for them to move through an environment which is fluidic, which is watery. So as a result, what will happen here, that in the intestine, what will happen that in the intestine, that in an environment which is watery, it is very difficult for the lipid to move from one part to the other because of the hydrophobic nature of the lipids or parts of the lipids. So the nature have devised a way here to circumvent, to deal with this problem, is that now these lipid constituents like free fatty acid, free glycerol and 2 MAG, 2 monoacyl glycerol, they will be then, along with the bile salt, they will be then contained in specific structures known as micelles. These micelles are basically arrangement of lipid in such a way that if we see the hydrophilic portion of these lipids is towards the liquid part of the environment in which the lipids is contained or in which they are moving, and the hydrophobic is directed inwards toward to make the inner core. So what will happen in this way, because of this reason that hydrophilic portion is interacting with the water, so what will happen, that the lipids can move in this environment, and it contributes towards the absorptions of these lipid molecules in a very specific cellular structures known as enterocytes. They are very specific cells, which are cells in the intestine, which are specialized for the absorption of the lipids and other molecules. So if we see again here that these are your lipid molecules, they are after the process of emulsification, they are converted into or package themselves in the form of micelles, and they then can move through the walls of the the enterocytes for the process of reabsorption, or for the process of rather absorption. The border of the enterocytes, also known as a brush border membrane, they can contain many microvilli, which actually contributes towards the absorption of the molecules. So the long chain fatty acids are then and relatively medium chain fatty acids, and the molecules which cannot move freely in the liquid, they are then transported and absorbed by the formation of micelles. But cholesterol, very small portion of the cholesterol is absorbed, around only 5% of the cholesterol. So in this way, cholesterol make an exception here. As far as the short and relatively small length fatty acids are concerned, they don't need micelles because they are not interfered by the water environment to the extent as the long chain ones are affected. So they don't need micelles, they can simply be diffused during this process of absorption. Then once now lipids have been absorbed into the enterocytes, what will happen now? And they have dealt with the environment because of the formation of micelles. Now again, what will happen? That their constituents from which these lipids were actually made up, they will be combined again, and again they will be ready for their final catabolism in the liver. So if we see here, so what will happen that as I have been saying again and again, that the major constituents of these different lipid molecules, for instance, the constituent of the triglycerides, that is the 2-monosylglycerol, it is converted into triglycerol. Again, triacylglycerol, again, because of the action of the acyltransferase enzyme. And similarly, the long chain fatty acids are converted again into fatty acyl CoA through the activity of fatty acyl CoA synthetase. And cholesterol, free cholesterol, is again esterified by the cholesterol acyltransferase, and you get cholesterol ester. Fine. Now again, you get these lipid molecules in their intact form. Now, for the movement of these lipid molecules towards the liver for their final catabolism, what is should happen here is that, you again see, let's say if they are moved through the bloodstream, again the problem will be facing, these molecules will face that their hydrophobic nature will not allow them to move. So what will happen then? Here they will be first be packaged into another very important molecule, that is the next important structure, that is chylomicrons, very important lipoprotein molecules. And these lipoprotein molecules then contributes towards their transportation for their towards their final destination. So in this way, you can see that how nature have devised different ways for the movement of lipid molecules from one part of the cell to the other, or from one part of the tissue to the other, because of the reason that lipids have a very hydrophobic moiety within them. So and most of your cellular environment is of course fluidic, it contains water. So in order to deal with this, nature had devised different ways. In certain times it makes micelles, on other times it makes chylomicrons, and in this way the lipids are finally, they move towards their final destination for their final or catalysis.